Hello there, it's Dr. Ominde. My last lecture, it was cut short at the spleen when we're talking about the immune tissue. So I'm just going to wind up on the histology of the spleen. So the spleen is a lymphoid organ that's located on the left upper quadrant in the abdomen, and it's mainly supplied by the splenic artery and vein. And the splenic artery comes from the celiac trunk. So what are the three main functions of the spleen? It produces immunological response against blood-borne antigens, yeah? So you're able to respond to microbes that are within the blood, and it removes particulate matter within the blood and also aged or defective blood cells. The spleen in a normal fetus or in adults with certain diseases is able to carry out hemopoiesis, is able to help with the formation of blood. What's the general structure of the spleen? It's covered by a capsule. This capsule is a dense connective tissue, and from it we have trabeculae that move from the capsule to go and divide the parenchyma of the spleen into incomplete compartments. So the large trabeculae originate at the hilum, and together with the neurovascular structures at the hilum, the branches of these neurovascular structures are carried into the parenchyma of the spleen following these trabeculae. The pulp of the skin or the parenchyma of the spleen has reticular tissue that contains reticular cells, blood cells, macrophages, antigen-presenting cells, and lymphocytes, which are part of blood cells. So the splenic parenchyma is made up of two parts, the white pulp and red pulp. White pulp consists of periarteriolar lymphatic sheath, pulse, P-A-L, periarteriolar lymphatic sheath, pulse, and lymphoid nodules. So these form the white pulp. So periarteriolar lymphatic sheath is just T lymphocytes aggregating around the central arteriole. Then we have the red pulp, the other component of the splenic parenchyma. This consists of splenic cords known as Billroth's cords and blood sinusoids. Sinusoids are just venous, endothelial-lined blood channels. So this is what we are referring to. The spleen as an outer capsule that sends in trabeculae to divide the gland into incomplete lobules. And the parenchyma, or the pulp of the spleen, is divided into white pulp and red pulp. The white pulp is a central arteriole surrounded by T lymphocytes, central arteriole T lymphocyte, central arteriole T lymphocyte, periarteriolar lymphatic sheath, together with the lymphoid follicles around them. Those form a white pulp. Then the red pulp is by splenic cords, the Billroth cords, and sinusoids, which are endothelial-lined channels of blood. So the white pulp is formed when the splenic artery comes from celiac trunk. It gets to the spleen at the hilum, then divides into trabecular artery that follow the trabecula, which are going to divide the gland into incomplete lobules. So these trabecular arteries will form arteries that will be enveloped by sheath of T cells. So when you have the central arteries that are enveloped by sheath of T lymphocytes, they form periarteriolar lymphatic sheath. So these central arteries are the white pulp arteries. They are surrounded by T cells to form periarteriolar lymphatic sheath. So they receive lymphocytes, mostly B lymphocytes, that will surround the periarteriolar sheath, and those will form lymphoid nodules. Okay, so a white pulp, central arteriole with T lymphocytes around it and B lymphocytes forming a lymphoid follicle, will contain marginal zone where you have the lymphoid nodules, and it has many sinuses and lymphoid tissue. This marginal zone also has few lymphocytes and many active macrophages. After leaving the white pulp, the sheath of lymphocytes thins out, and the central artery therefore forms straight peninsular arterioles. So from splenic artery to trabecular artery to central artery, which form peninsular arterioles. So the T lymphocytes around the central artery form a periarteriolar lymphatic sheath before they thin out and form peninsular arterioles. This is splenic artery gave this trabecular artery. Trabecular artery gave out a central artery. Central artery is surrounded by T lymphocytes. So this is your periarteriolar lymphatic sheath. Around it you have aggregation of B cells, and this is what is forming our lymphoid nodule. And then this nodule now has marginal zone. You can see the marginal zone where you have the sinuses, endothelial-lined channels of blood. Okay, this is our periarteriolar lymphatic sheath, T lymphocytes around central arteriole. Okay, and these are marginal zone sinuses. This is the germinal center of our lymphoid follicle. So this is aggregation of B cells. In the peripheral white pulp, you have B cells, B cells. Here we have T cells. And then these usually thin out and form your penicillar arteriole, from central arteriole to penicillar arteriole. So splenic vasculature, the splenic artery will branch into trabecular. Trabecular will give central arteries. Around central arteries, you have T cells to form periarteriolar lymphatic sheath before the central arteries thin off to form penicillary arterioles that will terminate into sheathed capillaries. So the sheathed capillaries usually lack endothelial lining and are surrounded by aggregates of macrophages. So these ones will form the first part of the filtering mechanism. Now, splenic artery, trabecular artery, central artery, penicillar artery into sheathed capillaries. So we have what you call open and closed circulation in the spleen. When blood enters the parenchyma of the spleen from the sheathed capillaries, it will be squeezed through the sinuses to drain out of the organ via the splenic vein. And that's where we are having the open circulation. So the open circulation of the spleen is where the blood enters the parenchyma of the spleen from the sheathed capillaries, and then through the capillaries into the sinuses, and then from the sinuses they drain out of the organ via the splenic vein. So this is open circulation from the capillaries into the parenchyma, then from the parenchyma squeezes through back to the sinuses, from the sinuses to the splenic vein into the blood circulation in the body. So that is open circulation. So it's a circulation from the blood channels. Okay, remember we say it's splenic artery, then from splenic artery you have trabecular arteries, central arteries, to penicillar artery, then sheathed capillaries. So from all these endothelial lined channels, the blood now enters the parenchyma. So that's where we are calling it open circulation. It enters into the substance of the gland before it goes back to the endothelial lined sinuses into splenic vein. So this circulation in the parenchyma is the open circulation, and the circulation within the endothelial lined channels is the closed circulation. So the red pulp of the spleen is made up of splenic cords and splenic sinusoids. The splenic cords are the Billroth's cords. And the red pulp is made up of reticular cells supported by reticular fiber. Then which other cells are in the red pulp of the spleen? We have macrophages, plasma cells, T and B lymphocytes, and we also have red cells, platelets, and white cells. So this red pulp is usually composed of splenic cords with these cells, and these cords are separated with irregularly shaped sinusoids, splenic sinusoids, endothelial lined channels of blood within the parenchyma of spleen are the splenic sinusoids. So the red pulp usually has incomplete basal lamina surrounding the sinusoids, and the spaces between the endothelial cells are usually small so that only flexible cells are able to pass through. The lumen of the sinusoids in the red pulp are usually very narrow, and the splenic cords are infiltrated with the red blood cells. So this is what we are referring to as number 1 are the splenic sinusoids, and number 2 are the splenic cords with the immune cells and the blood cells. These are the endothelial lined channels of blood, the splenic sinusoids. And this is how they appear under an electron microscope. These are splenic sinusoids and these are splenic cords. So this we have discussed the differences between open and closed circulation of the spleen. Closed circulation is just within the vessels. Splenic artery, trabecular artery, central artery, peninsular artery, sheathed capillaries into sinusoids and back to the veins. That's closed circulation. But open circulation from the sheathed capillaries, they enter into the parenchyma, so into the splenic cords before you get back to the sinusoid. So open circulation opens up into the parenchyma. Closed circulation remains within the blood vessels or endothelial lined surfaces. So basically, those are the differences between open and closed circulation. So from the sinusoids, the blood usually goes back to the red pulp veins that join to form trabecular veins, which will eventually form the splenic vein and back into the circulation. Remember, trabecular veins lack muscle walls, so They are just channels of hollowed out trabecular connective tissue that are lined with endothelium. So why do you need to understand the histology of the spleen? Because it is usually, it can be removed when there is splenic injury, when there is splenic rupture, in cases of anemia where the spleen is destroying a lot of red blood cells, you can remove the spleen. Then in cases where, um, let's say, for example, there's an infection, in cases where you've removed the spleen, in this, like a patient who has anemia or an abdominal injury where the spleen is bleeding, so you have to remove it. These patients are usually at risk of infection. These are what we are calling splenectomized individuals. They are at higher risk of infection after you've removed the spleen, depending on what was the cause of the splenectomy. Then we also have tumors of the immune system that may involve the spleen, such as lymphomas, okay, and leukemias, tumors of the blood. So this can involve the spleen. And another one, in lymphoma, it could be systemic or localized. So systemic will affect, localized will affect a specific lymphoid organ like the spleen, yeah? And these conditions may occur also in immunosuppressed individuals, such as in AIDS and organ transplant patients, or in certain infections. So in these cases, you may see the involvement of the spleen. So thank you very much.